Thank you, Mendy. Thank you, Rabbi Solish. It's a pleasure to be here. I'm actually incredibly flattered that there's so many people here. And delighted to be here. Must be a slow sports night. But I'm thrilled to be here. I'm very excited to talk about this field that I just was calculating. I've been involved in this field of genomics for more than half my life, and I'm in my mid-60s. So you can quickly do the arithmetic and find that it's been a while. I'm always really enjoy talking to new audiences and including audiences that are eager to learn. And if you're here tonight, it must be because you're really eager to learn about genomics. So, but before I get that, I do want to set a context of who I am and why, but one of which is that for the last 30 plus years, I've been working at the National Institutes of Health, part of the US federal government, part of the executive branch. That is one of the iconic buildings that you can see on the left. And on the right is an aerial view of the NIH in Bethesda, Maryland, where I grew up and raised my family. And what you did come across when Mendy was here, part of the connection here is that my son Josh is here, is a classmate of Mendy's. Now, my son's now a fourth-year medical student on the MD track. Mendy's on the MD PhD track. But that's how I got to know Mendy. That's how I got connected. And this is a sort of reason that I have now been in the Washington, D.C. area, living in Bethesda for 30 plus years. I even met somebody, by the way, already tonight who was born at what was then the Navy Hospital, but is now Walter Reed Hospital, just across the street, just on the lower part of that, just across the street from the NIH. In terms of what the NIH is, it consists of 27 components. I don't want to get too geeky, but there's 27 parts. I biased way put us in the center of the universe there in the middle, the NHGRI, National Human Genome Research Institute. But you know, there's that cancer. Most of the other institutes are involved in specific diseases or to specific disease areas, Cancer Institute, the Eye Institute, the Deafness Institute, Neurology Institute, et cetera, and there's 27 of them. But then each have their own unique story. Our story is one where we were created by the US Congress just before 1990. So I do have things to share with you for you to take home. And one of them is this brochure. So on your seat was a brochure. There's a couple things about the brochure I'm going to tell you about. But the first thing about the brochure is it's a very nice overview of our institute, what we do, some of the science I'm going to tell you about tonight. And it really is a very nice, easy read that as a background, I hope maybe you'll take a look at if you enjoy hearing what I'm talking about tonight. Also, though, there's fun stuff in there because if you go to that back pocket, we stuffed some things. One thing I'm going to talk about later, but this is actually brand new. We have a great tchotchke to share with you. We have these pins that we made recently, which are the rage. And so you get one of these. So take the pin, and if no one's sitting next to you, grab theirs too and take that home. So I always grew up in an environment that if it's free, you take two. And so grab two. In any case, so that to tell you a little bit more about the Institute, but you know, I don't want to really spend a lot of time talking about government bureaucracy. It's a crazy week in DC. I'm so glad to be out of DC for a little while as this massive transition takes place that will have sort of effects on us at all parts of the government. But for now, I'm having much more fun being down here, talking to all of you. But what I want to tell you about is genomics. And some of you may have come here thinking, either before or after having a drink, you know, why should I care? You know, why should I care about genomics? Well, I will tell you in my very biased way that genomics is scientifically weightful, and I will show you this. It is associated with some remarkable history of exemplary achievements. I'm going to tell you about the biggest one being the Human Genome Project. But most important, which is why I'm here, is because this is becoming increasingly relevant in everyday life around the world. And in particular, it is now relevant to human health and the practice of medicine, which means it's relevant to all of us, all of our families, and all of our friends. And so what I will convince you of, I think by the end, is just be prepared to be convinced that you should care about genomics. And if I don't successfully do that, then I certainly would have failed in my mission. But I also realize that there's a very heterogeneous audience. I have really intimidating folks like medical students here that know a lot. And then I know I have some physicians and scientists in the audience. And then I have others who come from different backgrounds. And so I realize knowledge of all the intricacies of DNA and genetics and genomics is probably highly variable. So, but I'm going to really try to bring everyone along. So I'm not going to get too geeky or nerdy, but I'm happy to get geeky and nerdy during the Q&A session. But I really want to make this as accessible as possible to as many of you. So let's just start with a very simple question. And that is, what is the blueprint that is used to build and operate one of these, which happens to be a little relative of mine, okay? That is a fundamental question that we have wondered about for centuries in some ways. What is the blueprint of all of life forms? Not just critters like us, but all life forms. Well, I will tell you that there were two scientific fields that got launched last century that have really brought great clarity to that question, finally, even though we've been thinking about it for a century. One was in the early part of last century was the creation of the coining of the word genetics. And actually that didn't exist until 1907 as a word, and it created a new discipline. Now, there's lots of ways to define genetics. Think about it as, I think most simply as it's the study of inheritance, how traits get transferred from one generation to the next, one cell to the next. When genetics got coined as a word and the field got created, we had no idea what was the blueprint material that was inherited. We had no idea. It took a series of discoveries throughout the last century that at the midpoint of the century led to the elucidation of this iconic structure, this famous double helix, which you see everywhere constantly, which is really the structure of DNA. And I'm not even going to go through the structure other than to say this was sort of the final key piece of the puzzle, that DNA was that blueprint material. And then there were a series of remarkable advances that took place throughout the rest of last century that led in the late 1980s to the coining of the word genomics and the creation of a new field of genomics. And the key aspect of all of this is that DNA is basically the blueprint material necessary for all of life. And so let me just give you a few more concepts before I bring this all together to really tell the main part of my story. So all living systems consist of cells, essentially. Each of us consists of somewhere on the order of 30 to 40 trillion cells, human cells in our body. And every one of those cells, every one of them operates off the identical blueprint. And that blueprint exists in the center of the cell, a structure called the nucleus. And it turns out in the nucleus terms that in the nucleus are these structures called chromosomes. Let's make it simple. Think of chromosomes as suitcases that are responsible for carrying the blueprint information from one cell to the next, from one generation to the next. But the key aspect of chromosomes in the suitcases are DNA. DNA is the blueprint material for all life forms, and every one of our 30 to 40 trillion cells in our body operate off that same exact DNA blueprint that each of us got from our parents. So a genome is all of the DNA of an organism, okay? And so the human genome consists of this series of suitcases. Turns out that the human genome, we have 46 chromosomes. We got 23 from mom, we got 23 from dad. But they're actually the same sort of, they're similar copies, but we just got one set of suitcases from each parent for a total of 46. And of course, each of those chromosomes consists of this DNA. And here's one last scientific fact before the fun really will begin. The scientific fact is that DNA consists of 4 building blocks. It's 4 chemicals. I'm not going to tell you the chemical names. We'll just use the first letter. One of the chemicals starts with an A, one starts with a G, one starts with a T, one starts with a C. But there's a precise order of those nucleotides, and that is known as a sequence. That's the code. That's the blueprint. It is the order of the letters in DNA that mysteriously encodes all the information necessary for creating and operating any living creature, including the human being. And the genome's big. I guess my understanding is so is the Talmud, but we'll come back to that. But genome's big, right? I mean, it's 3 billion, it's 3 billion letters. It's 3 billion of these building blocks, G, A, T, C, C, A, and 3 billion across all those 23 chromosomes, one set of those suitcases. That is what the human genome is made up of. So fundamentally, what is the blueprint that's used to operate one of these? I'll show you exactly what that blueprint is. It's 3 billion letters that look just like that, which is really pretty astonishing when you think about it, because it's not exactly a good read. I mean, it's only 4 letters. You don't even get spaces or capitalization. That's just it. But 3 billion letters of code, which you can represent just like that, is all that takes to create and operate one of those. Well, 3 billion was a big number, but it wasn't an infinite number. And so the idea started to come is, well, if we could get really good at studying DNA, can we ever get to the point we can actually just read it all out, figure it out, have the blueprint in front of us, or at least on a computer? Well, it turned out that throughout the last century, especially after the discovery of DNA's double helical structure in the 1950s, came a series of discoveries that just accelerated progress in DNA research. Don't worry about the details, but in the 60s, we figured out some of the code, the interpretation of how it was that DNA encoded the information for making cells and making bodies operate. By the 1970s, we learned how to read the letters of DNA. That required a lot of chemicals and fancy innovations in technology, but we could figure out how to actually read out the letters of DNA. And then in the 1980s came the ability to clone and manipulate DNA and be able to propagate it in the laboratory, and set us up in a circumstance that it began to make sense that maybe, just maybe, we could mobilize and accelerate all of these new technologies starting at around 1990, and once and for all, read out the 3 billion letters of the human blueprint, otherwise known as the human genome. That, my friends, was known as the Human Genome Project. So this is where I come into the story, because I was actually at the other end of what Mendy's trying to do right now. Mendy's trying to get an MD PhD. He's not trying, he's going to get an MD PhD, but he's halfway through or something like that. And when he's done, he'll be a physician scientist. He'll have both a degree as a physician, as a degree as a scientist. And that's exactly what I was. And then I got an opportunity as I started to train in pathology to go into a research lab. And there was this cool new thing called genomics. But I didn't tell you one fact. I graduated medical school with my MD PhD in 1987, the same year the word genomics was coined. So by the way, if you're impressed with me, just remember, I never heard the word genomics once in all of my formal education up through my MD and my PhD, which may mean that I'm a fraud because I have really no formal education in this very field. And yet, to be honest with you, I'm sort of the genomicist in chief of the United States. So it sort of either tells you that this is really a wacky system, or it tells you, especially the young people in the audience, you're a lifelong learner. You can throw as many degrees after your name, but you may still become an expert in saying that you never heard about when you were getting any of those degrees. And that's absolutely my life story, because I got involved in genomics in a field I never heard the word once in all of those years that got me to an MD and a PhD. So it's just a side note. I was fortunate. I was a freshly minted physician scientist, got involved in the Human Genome Project literally on day one. But I can't stress enough how unusual this project was. Most of biomedical research up until that point was done by individual laboratories having great thoughts and thinking about hypotheses, and then groups of 10 and 20 researchers would work together, just that one lab, and they would do things. That wasn't the Human Genome Project. The Human Genome Project was bringing thousands of scientists from around the world together, multiple countries, multiple funders, and basically not have great thoughts, except like, how the hell are we going to do this? This was the most profound thought we had, because we had really no idea how we were going to scale everything up to be able to read out 3 billion letters. And yet off we went to do it. And I'm going to basically just tell you, without giving you much of any detail, was that we did it. I mean, after 13 years of incredible efforts that involved many, many scientists, many, many skeptics along the way, and many, many hurdles, we eventually figured out how to do it. And some of you may remember this, or some of you may have read more about it. But at the end of the day, the Human Genome Project actually was successful. We accomplished all of our goals, and the signature goal was, for the very first time, we read out the 3 billion letters of one representation of the human genome. And that was incredible, and all that data got shared, and we put it up on the internet, and the rest is history. And that was in 2003, and now we're in 2025. Now, sometimes I find it really useful for especially folks that haven't thought about this much, about what does 3 billion letters look like? Like, when I say the human genome, like what does that really look like when you have 3 billion letters of code? So let me give you a couple different metaphors. So here, one metaphor is you're making a book of the human genome sequence, and you go to do that. So you could just type out the 3 billion letters, the G's, A's, T's, and C's, the human genome. And if you did it with no spaces and 12-point font on 8.5 by 11 sheets with standard margins, it would require about 1.2 million pages. Okay. So pretty remarkably big book. So here you can see, I mean, this is more complicated than 12 minutes. You know, I only get 45 minutes to tell you about my book, but I heard there's like a 6-part series and you have to go lecture. That doesn't seem fair, but that's me reading the human genome. Okay, at least according to the artificial intelligence tools that we can use. But that's pretty incredible. But here's another one. There may be some computer nerds in the audience, certainly I'm one. So I'll give you another metaphor that I think is also pretty remarkable. If you go to store a human genome sequence on a computer, if you actually did one of your genome, remember you actually have 2 genomes. I sort of remember one representation of a human genome is 3 billion letters, but any one of us has 6 billion letters because we have one genome we got from mom and one genome we got from dad. But so a person's genome is 6 billion letters, okay? And if you put that on a computer, it takes about 1.5 gigabytes. Oh, it's misspelled, but it's gigabytes, it needs a T, and that would to store it, right? Now, I'm doing this presentation in PowerPoint. You may ask, how much space do you need on a computer to load and operate PowerPoint? And it turns out it's not that much different, right? It's just a little bit more. You actually need more computer space to run PowerPoint than you do to represent your entire genome on the computer, which is sort of really remarkable when you think about it. And yet that's all that's needed to basically go through and operate one of these things. Is actually, you know, the fact of the matter is the code for a human requires less computer storage space than the code for, you know, I mean, that just blows my mind. So I just have to share that. Okay, so that was the key thing, but that only brings us to 2003. And what happened since 2003, this is where the story is going to get fun, is because while I could speak for hours about the Human Genome Project, you know, it was both the finish line, but more importantly, it was the starting line. It really became the starting line of what I'm going to tell you about now. Because, you know, getting the sequence was one thing, but that was not just what we wanted. We wanted, and especially as a physician scientist, we were extremely motivated to move on with that capability and with that knowledge to do incredible things. Now, there are lots of speakers you could invite after me if you want to learn a lot about how genomics has found its way into all sorts of way cool things. And every one of these could be a story in and of itself, a speaker, I mean, it's a whole field. Every one of these things are genomic applications in those areas are absolutely remarkable what's transpired. But I'm going to keep it focused on health and disease and medicine, partially because that's what I do. I work at the National Human Genome Research Institute, and this is all part of the National Institutes of Health. So everything I'm going to tell you about now is how 23 years ago, we pivoted and really started thinking about, at our institute in particular, which had been created exclusively to help run the US's contribution to the Human Genome Project. But now, starting in 2003, we needed to show why it was so critical that we completed the Genome Project by thinking about how to improve the practice of medicine. Now, one of the reasons why the US Congress bought into the idea of the Human Genome Project to fund it, and by the way, I didn't give you a dollar. The whole Genome Project cost about $3 billion across multiple funders. I mean, $3 billion, it's, you know, it's a lot of money, but on the grand scheme of things, not that much money. Moonshot cost $25 billion, or something like that, to put it in context. But what Congress liked about it, at least in the US and other countries funded this as well, was we told them that too much of what we learned in medical school, and even much of what they're learning in medical school now, is based on treating the average patient. Patient comes in, they have to figure it out, and then you figure out how to treat them. But no patient is average. Every patient, including every one of us, we're all unique. And we bring different things to our uniqueness. Some of it is our physical and social environment, our life experiences, exposures, and so forth. But some of it is also our blueprint, because each of our blueprint is just a teeny bit different from one another. And there are many features in our blueprint, differences in our DNA, that make us more or less susceptible to diseases, or make it circumstances in medicine where you could be a little bit more customized in how you approach it. So we always said, we're flying blind as physicians because we are taking care of people with no understanding of their specific blueprint, even though every one of us carries a unique blueprint. The Genome Project would give us baseline information about that blueprint, and then eventually we could use it in some meaningful way to be able to go through and be able to have the ability to use genomics in the practice of medicine. With that in mind, it just should come as no surprise that the popular press, like New York Times Magazine, not to mention the scientific press, shortly after the Genome Project ended, almost immediately began 2 words of genomics and medicine. It was the logical thing to do. It was the promise. We were in a new journey, and it was very clear that one of the new aspects of the journey was to get genomics infused into the practice of medicine. So that was wonderful. We appreciated it, and we knew how to spell genomic medicine, and we had absolutely no idea what it was really going to look like. It was a very blurry concept, and we had just finished the Genome Project. We didn't even understand what all those letters meant, and somehow we were going to change the practice of medicine. And it became very clear, though, that that had to be the new finish line. Now, we can define it, and it basically come up with various definitions, but it's basically using genomic information about a patient to tailor their medical care, to not treat everybody on average, but eventually get to the point where we could have information about a person's differences in their genome that might be influential or consequential for their health and for how we might want to treat them medically. Now, by the way, I will tell you, you'll hear of other terms. I'm sure you have heard other terms like personalized medicine, individualized medicine, precision medicine. Those are all great terms. I don't disrespect them. They tend to be terms that go beyond genomics. So, you personalize it based on various things, or you individualize it. That some of that might bring in non-genomic factors, all important. I'm just going to stay laser focused and keep using the phrase genomic medicine because my expertise and the area that we focus on are the genomic aspects of individualizing medicine. So I will call it genomic medicine, but think of these other terms as slightly broader concepts of something very similar. So it's a journey. The Genome Project was a journey. We went from the beginning to the end. Now we got a new journey. It's just the starting and finish line flipped. Now we have a new starting line. The completion of the Human Genome Project was once our finish line. It's now the new starting line. The finish line is changing the practice of medicine. And just like the Genome Project, we knew what some of the steps are going to be, but we don't know what all the steps are going to be. And just like the Genome Project, you just got to get going and you figure out what step needs to be done. And when you complete it, you check it off your list and you just keep soldiering on. And when you hit obstacles, you figure out how to overcome them. And so this is the journey that began with this grand pivot in 2003. And it's a journey that really has seen some truly remarkable advances. Now, the challenge of giving a talk like this in 45 minutes is I occasionally have to compress 22 years of progress into about 3 minutes or 4 minutes. This is going to be one of those slides. So I want to get to some of the medical applications in particular. So I'm going to go and just tell you some highlights of what some of these steps were, but I'm going to be very, very high level, but please ask me questions if you have them when I'm done, or afterwards. I'll happily answer questions for as long as people have them. So the first thing I can tell you is that you got to deal with the first step. And it was very obvious when the Genome Project ended in 2003, what the biggest barrier was. And it was quite simple. It was too expensive. So we succeeded in sequencing the human genome, but it cost, the sequencing part of it cost about $1 billion, okay? It was a great investment of $1 billion, but remember, I want this to be a clinical test. I want a patient to walk in the door, we want to sequence their genome. We can't, $1 billion is a little expensive for a clinical test, right? And so we said it needs to be $1,000, okay? And guess what? Fast forward and incredible advances, we did it over the last 22 years. We've reduced the cost of sequencing a human genome over 1 million-fold. You can get it done now for less than $1,000. You know what you get to do when that happens? You check it off your list. You say, done. Well, now what? Well, okay. Well, we sequenced one representation of the human genome. Well, that's great, but actually we're more interested in how we differ than how we're all the same. Now, it turns out that we always knew we were over 99.6 or 5% the same, but we were really interested in those differences because buried in those differences are lots of things that are consequential to our health. In order to figure out what's out there and how we vary, you got to sequence a lot of genomes, and then you got to catalog the differences. We only had one in 2003, one genome, not even, we had half a human genome, it was one representation. So what we've done, We've now gone out, we've sequenced millions of genomes because they got cheap. And you know, we went from having knowledge of maybe 30,000 places across those 3 billion letters where we know people vary. Now we know of hundreds of millions of places across those 3 billion letters that we know people vary. We have rich databases that talk about this variation. We have it from different populations, different ancestral groups, increasingly from populations all across the world. We are building up lots and lots of knowledge about how what's known as human genomic variation, how the DNA of humans varies across the world. And eventually we're going to figure out what all of that means. But billion genomes or more, so we can cross that off our list. Lots of knowledge about genomic variation. But of course, if you know you have sequence changes, if you have spelling changes, you got to know most of it is actually completely inconsequential. A subset of it is actually biologically consequential, but maybe not medically consequential. And a very small subset of the differences are actually medically consequential. To figure that out, you have to know how the human genome works. We know the 3 billion letters. We do not know when the Genome Project ended what any of that interpretation was, but we absolutely knew we need to figure it out. And so we've gone a long way between figuring out what that code really means. I'll give it a medium check mark because I predict that my children, my grandchildren will be interpreting. I mean, it probably is like the comment. I mean, we will be interpreting the human genome sequence for generations to come because it's really complicated. In fact, it deserves a 10-part series, not a 6-part series like we're talking. So it's really complicated. But we've made a lot of advances. By the way, I suddenly realized I want to give you a number back on when we're talking about variation. Look to the person to your left or look to the person to your right, and I will tell you that across your 6 billion letters, there's probably about 3 to 5 million letters that are different between you and that person. So you may say that's either a lot because it's 3 to 5 million differences in your gene in your blueprint, or you may say it's a little because only about 99.6% or it's 99.6% identical. It's a very small percent difference, but still 3 to 5 million letter differences are quite a few. But of course, up until then, it didn't really say anything about human health or disease. But with the ability to sequence genomes and with the ability to have understanding about how our DNA varies, with the ability to understand how the human genome works, we started to look at people with diseases, especially the genetic diseases, of which come in various flavors. Some are more rare and some are more common. The more rare flavors, such as cystic fibrosis, Tay-Sachs disease, sickle cell anemia, Huntington's disease, those are pretty simple to figure out, it turns out, because it's usually one functional part of the genome called genes, but we don't have to worry about the name. It's just one functional part of the genome that's broken because of a typo. Think of it as a mutation, a DNA change, a typo. It breaks that functional component and you get sick. Turns out that's gotten fairly straightforward to figure out because we now have the ability to figure it out. And so how do I know that? Because when the Genome Project started in 1990, by the way, there's thought to be about 10,000 rare diseases, of which I just named a few, like Tay-Sachs and sickle cell and Huntington's and cystic fibrosis, about 10,000. When the Genome Project started in 1990, there were 61 of those that we actually knew what was broken. Only 61. Now we're coming close to 7,000. That's incredible. We now know exactly what is broken in people with these rare diseases for roughly nearly 7,000 of the 10,000. That's incredible progress. And that's remarkable. However, I will also immediately point out that rare diseases are not they're incredibly in aggregate, they're quite common because there's a lot of rare diseases, but they don't fill hospitals and clinics around the world. They're devastating to families, of course, and to patients, et cetera, et cetera. But healthcare burden-wise, most diseases, hypertension, diabetes, cardiovascular disease, are what are known as common diseases. And those are not where one functional part of the genome has a typo, but rather there's many parts of the genome that have smaller typos, and they all sort of conspire together with what is typically a greater contribution of the physical and social environment. And those were making progress, but the progress is slower. It's much more complicated. So bottom line is, though, we've made significant advances, but just like, once again, this is going to take many generations to fully flesh out. We've done really well with rare diseases, and we need to do better with common diseases. So again, I give it a medium check mark. But now what I really want to emphasize in this part is is this pivot to medicine, because nothing about those first 4 highlights says anything about medical advances. But we are there. We have started to see genomic medicine come into reality. And I'm only going to give it a little check mark because it's just the earliest examples. And it only started about 7 years ago or 8 years ago where I could start to give these examples. But it really is remarkable. And again, I'm just going to briefly unpack my highlight reel here. But every one of these is a story in and of themselves. So we have gotten to the point that here and now, we are starting to see genomics being used in the practice of medicine. In one area, I'm going to give you about 5 areas. One is cancer. Turns out, what is cancer? Cancer is a disease of the genome. Every cancer are examples where DNA has picked up typos, many of them, making those cells grow out of control. And cancer, now, but we can sequence the genome of cancers and learn a tremendous amount about that exact cancer in that exact patient. And it's just totally changing oncology completely. Not yet for all cancers, not yet routinely in all settings, but I will absolutely go on a ledge of saying that I think 50 years from now, the greatest contribution of genomics in medicine will be in cancer, because I really think this is just game-changing. It's just a perfect circumstance for genomic medicine. Second area is, I was hinting at this, is rare genetic disease diagnostics. It turns out that nowadays, the child presents or a young adult presents with a suspicion of having a rare disease. More conventionally than previously, they work up, spend tens and $20,000 and $30,000 worth of workup at a much earlier stage nowadays across not only the United States, across the world. You'll just sequence the genome of that child. About 30 to 50% of the time, they'll figure out what's wrong with them. And it'll eventually get higher, but because we'll learn more about the genome. But it costs less than $1,000 to do it, or even if it's a couple thousand. It's much cheaper on workup. It is absolutely here and now. And it's happening tens of thousands of times every year in the United States, probably double or triple that around the world. There's a couple of sub-stories of that I always like to point out. First of all, there are people that and you may know some of them, who have for years, often adults at this point, have gone around with what are called undiagnosed conditions. They have a disease that nobody has ever been able to figure out what is wrong with them. And basically, these are often people that go from doctor to doctor, medical, different medical centers, sometimes different parts of them. They'll just go anywhere. They're desperate. Their diagnostic odyssey is painful. It's horrific. And what's happened is genomics has swooped in and been incredibly helpful. There's now programs around not just the country, around the world for undiagnosed diseases programs where experts come in. They really work these people up clinically, but they also use high-tech genomics. They don't always figure out what's wrong with them, but they're doing better and better all the time. And there's enough success stories to make this an incredible, productive program. The other sub-story of rare genetic diseases, which is overwhelmingly heartwarming and absolutely is going to become mainstream, is if you go to any neonatal intensive care unit in the world, there will inevitably, especially the big one, or ones here in Atlanta and other places like that, where there are tertiary care facilities, there will be babies there where the neonatologist simply cannot figure out what is wrong with them. Sometimes we'll be able to say, this child will probably die within 2 to 3 days. There's nothing we can do. We've tried everything. We have no idea. We have now, over the last 5 or 6 years, worked out protocols through research that we supported, where they will sequence the genome of that child in less than 24 hours. And so far, about 30 to 40% of the time, and it'll always get better if they figure out what's wrong with them. And in many cases, you can intervene and save that child's life. You don't need to save very many babies before you realize that, holy cow, this is incredible. What's happening is more and more states are adopting regulations, policies, implementation of programs, and so forth to make it required that NICUs have, or neonatal intensive care units, have this capability of rapid genome sequencing. It's truly remarkable how this has become, and it's not just here in the United States, it's actually being implemented elsewhere as well. It actually turns out that the #1 genomic test here today is in the prenatal arena, something that a community like this, I'm sure, is quite familiar with, because prenatal testing has been sort of mainstream in Jewish communities, in my family, for example, and even with my 2 children. But the difference is that, and I bet you there's people in this audience that underwent prenatal testing through more invasive ways of accessing the fetal DNA, like amniocentesis or chorionic villus sampling. None of that's needed anymore to screen for the classic screening that you do prenatally, because now it turns out that these new methods we have for sequencing DNA are so exquisitely sensitive that you could take advantage of the fact that the placenta, which represents fetal, the DNA of the placenta is the DNA of the fetus, and little bits of DNA just naturally get shed from the placenta and float around in the maternal bloodstream. So with a simple blood draw, and the one thing I do know is pregnant individuals get lots of blood draw, so they're used to that. With one extra tube of blood, boom, you get this, and they analyze the small amounts of fetal DNA floating around in the blood, and you can do the same screening that used to require amniocentesis or chorionic villus sampling. 6 to 8 million pregnant individuals will get this non-invasive prenatal genetic testing now annually. Part of the reason why is because insurance companies started paying. You could have a whole different discussion about insurance companies, but the fact of the matter is, it started to get paid for because it was so much cheaper. There was no procedure. It was not invasive. By the way, my wife tells me that having gone 200 amniocentesis is not very fun. And they're a little dangerous. I mean, occasionally you can have a bad outcome. So the fact that you could do this non-invasively is just spectacular. So another spectacular example. One thing that's in the works but isn't quite there, why is it that every one of us either personally knows this or knows of family members or neighbors where they got a medication and it didn't work, or it made them sicker, or they need to change the dose, right? Let's talk about imprecision. I mean, the way we prescribe medicine is incredibly imprecise. Well, it turns out that we are now learning that a lot of the reason we vary, I mean, by the way, it's not because the physician's mean, and it's not because the pharmacy is incompetent. Every drug they give out works. It just doesn't work in everybody, or it works differently in different people. Turns out that those medications are metabolized differently by each of us, in part because of DNA differences that involve pathways that metabolize drugs. And we are learning more and more about which DNA changes indicate whether or not you should get a high dose or a low dose, or among the 10 hypertensive medications, which is the best one for you, or the 10 antidepressants, which is the best for you, or the 10 cholesterol-lowering medications, which is the best for you. It's selecting the best medication among a bunch of options based on DNA profiling. This will get more and more uptake. It has been a little slow. There's lots of reasons for it. Insurance companies have been slow. I think in the long run, though, don't be surprised if in the next 5 years, you go to your physician and they're going to say, I'm going to give you a medication, but first I want a DNA test done because we don't have one yet. So don't be surprised, and that's fine. Last area is not more so about medicine, it's more about prevention. And I just want to just briefly say that prevention, because sometimes people say, is it all about reacting to when you're sick? No, they really are getting to the point where, oh, I'm sorry. Oh yeah, that's right. It is. Let me go back. I'm sorry. I don't have that slide. There's really two arenas. Well, one arena I want to tell you about. We are starting to do prevention. And some of you may know, I mean, some of you may be familiar. If you have as one good example, often it's based on family. You may have a family member who gets breast cancer at a young age, gets colon cancer at a young age. And we are now increasingly getting to the point that when you hear about that, they say, tell all your family members, go get tested for DNA, because there's a number of syndromes. And the classic, of course, is the BRCA1 mutation. Angelina Jolie brought this to light in a very public way, but where we now know that if it's in a family, there's prevention that can be done. We can absolutely figure out if you have a BRCA1 mutation. We can figure out if you have a mutation in a set of genes that we know means that you are more prone to colon cancer. And if you know that's true, you can get screened, and we can prevent this from getting to be an advanced disease. Similarly, the reason we screen our children when they're born is we want to find out those who might have DNA changes that means they need to alter their nutrition. And if you don't alter their nutrition, they can end up with cognitive delay. That's another intervention based on genetics. So increasingly, you will hear more and more about prevention. It raises questions, which I'll just plant, and we could talk about in the Q&A if you want. You know, when will we reach a point that maybe we should just sequence every child at birth, sequence their genome, and have that part of their medical record? And that's an active area of discussion that I think will go on in the next, will absolutely go on for a long time, and we will see. Some countries are starting to contemplate that, and some countries are doing major studies to see. In England, they're starting to sequence large groups of babies at birth and trying to see, does genomic sequence information at birth help the health of that child as they traverse different ages and they get medical care throughout their life? So what I would say is, we really are celebrating this incredible arrival of genomic medicine. And before I end, I do want to, though, also plant a couple other ideas just to make it real. If you didn't notice already, I tend to be a little enthusiastic about what I do, but I don't want you to think I'm not grounded. And I also don't want you to think just because I have good PowerPoints and I tell a good story that any of this is easy. And what we spend a lot of time, as we should, at the National Human Genome Research Institute is we think about the challenge. I mean, we are all about knocking barriers down. We see what the benefits could be, but we don't want you to think that any of this is particularly easy. We still face some pretty big challenges. I could go on for days talking about all the challenges. I just plant a couple for you, just so you can appreciate the kind of things that do keep me up at night. The first thing is I did give you a bit of an oversimplification. I made it seem like we could just jump in and I could just, get some blood from any one of you, or I could get a cheek swab of any one of you. We can go down to Emory Hospital or down to CHOA, and we could sequence any patient who's there, and it would just boom like that. We would immediately know exactly what the case is. That is absolutely, just not the case. It is overwhelmingly challenging to analyze the patients. Even the child, we're doing this on 24 hours. It is still not a perfect system, because when we round on most of these individuals, and we have the list of 3 to 5 million letter differences in patient's genome, we don't know what most of it means. But we're getting better all the time. That's a big part of what my institute is supporting, is research to make this better. And there's lots of work going into this, and there's many aspects of it. But I don't want you I don't want to leave you with this impression that all this is push button. Eventually it'll get to be, I think, that, but it's probably a generation away. It is still a hard problem, but we are really good at cream skimming, and we get a lot of successful stories out of our work so far, but we still have a long way to go to make it routine. The other big issue that I do want to leave you with is that, when I started, when I was, that early days of the Human Genome Project, the community of genomicists, just a bunch of nerds like me thinking about how we're going to map and sequence the genome. And we really were isolated, and we didn't have to deal with societal issues because society barely knew we were around. And the analogy I make is that, during the Genome Project, I don't know what this is like the holiday meals at the kids' table. During the Genome Project, it was so much fun at the kids' table, because that's what genomics was for. We were at the kids' table. But what's happened more recently is, no, Eric, you can't sit at the kids' table. I don't care that they're talking about iPhones, Instagram, and Taylor Swift. You have to stay here to talk about mortgages, life insurance, taxes, politics, and genomic medicine and healthcare. I absolutely can tell you my entire community, and certainly I feel like we've made this transition, which we wish in some ways we didn't have to make. We're victims of our own success, but we're at the adult table now. And we take that actually quite seriously. We recognize that genomics, because it touched healthcare, it's now part of a societal set of issues that, we haven't been really perfect at solving. And genomics is not going to solve these either, but we're going to have to be part of the discussion. Because now all of a sudden we have to deal with who pays for all this. Is healthcare fixed in America? God, no. And but now all of a sudden, the biggest battles we're having is who's going to pay for this genome sequence, who's going to pay for the interpretation, who's going to pay for all this. And that we're just at that's an adult table problem. We have to deal with regulatory oversight. We didn't talk about regulatory oversight at the kids' table, believe me. We have to deal about equity. And believe me, we have major concerns that genomics could exacerbate health disparities if we don't figure out the payment and all the other things. We think a lot about equity. We need everybody to benefit from these advances. Lots of concerns about privacy. They're completely valid. And we need to ensure that our progress isn't hindered by a lack of attentiveness to genomic privacy, as it's true for all health data about individuals. And then we have lots of issues around literacy. Literacy for health professionals, literacies for the practicing physicians that are out there who are my classmates who never heard the word genomics once, but they're on the front line with patients coming in, already having their genome sequenced and saying, hey, doc, what does this mean? My classmates have no idea what it means. They didn't even hear the word genomic. But of course, we have to think about how do we train the next generation who are now in medical school? How do we think about all the other health professionals that it's not going to all be physicians. It's going to be a whole host of other medical professionals that have to help us. And of course, we need to think about everybody else, patients. because the language of medicine in the future is going to involve basic concepts of genomics. So we have to think about genomic literacy of the general public. And we're thinking about all these things, and those are all adult issues. And not to mention there are many, many other challenges as well, as you could imagine. So what I would say is what I get to do, which is a lot of fun, is I get to lead a community at what we now rebranded ourselves, the forefront of genomics, because everybody's doing genomics, and even all institutes at NIH now do genomics. We're sort of at the front edge of genomics, and we're at the forefront, and we always operate off of strategic visions that we publish about every 8 to 10 years. And so the last surprise in this packet of stuff is our latest strategic vision was published during the pandemic in October of 2020. And we published mostly typically in Nature. And so there in the back pocket is a reprint. You can see the title, Strategic Vision for Human Health, the Forefront of Genomics. I believe that having listened to me now for 45 minutes, you should read this. You will follow most of it. It is written in a way that I think is reasonably accessible. I think it will, and you'll get a sense. You'll see lots of things touching along the way, and a lot of things I didn't cover. So 10 pages in Nature, and I gave you this really nice, fun, glossy reprint. And so that's also what you can have. But I do want to end with one fun thing that I want to tell you. So we published these strategic visions. We did it during the Genome Project. We did it right after the Genome Project. We published several of them, and I put many of these, several of these I've been involved with. But we never did what I'm about to tell you about, is that we never sort of made hugely crazy predictions about the future. And when we were putting the final touches on this publication in Nature, and I think it was because we were a little stir crazy from the pandemic, because we had to finish it all by Zoom. We were all just like giddy. And so we said, you know what? And this is my staff and I at that point, because we had already engaged with the community, said, let's make some predictions about like what's going to happen 10 years from now. And so it just sort of caught on, and we said, well, that's a lot of fun. Let's do it. And so when you look at this preprint, we actually have it. It's right here. It's in box, whatever box number it was, box 5. And then the journal editors. I don't know, for those of you who are scientists or physicians, when you write us a paper like this, they always say, well, every display item, whether it's a table or a figure or a box, you have to refer to it in the text. So you just can't put a box in. You got to refer to it. And so they say you have to refer to the box. It's all right. So near the very end, we have a little paragraph, and then I said, all right, you got to say something about box 5. And so we said, but now more than ever, solutions to these problems seem to be within striking distance. And towards that end, with the characteristic spirit of genomics audacity, we offer 10 bold predictions about what might be realized in human genomics by 2030. Box 5. Box 5. So now we have the box, right? And then what happened? Every science reporter who read this, and a lot of scientists who contacted me, they didn't want to talk about the other 95% of the paper. They wanted to only talk about the bold predictions. And so I turned to our communications group. I said, you know what? That sentence in yellow wasn't adequate. We need to bring the bold predictions to life. Let's make a video. And so we did it. It's a very fanciful, fun video, but it'll get you thinking, is what I predict. So here's our video. There's supposed to be sound. There it is.
Genomics, which is the study of all the DNA in an organism called genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward to the success of the Human Genome Project, a massive reduction in the cost of sequencing the human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future. A future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully obtained by 2030, achieving even one or more of these by that time will have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5. Student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6. Genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7. It will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8. A person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9. Advances from human genomics will benefit all of society, not just a few. Prediction 10. Genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020sd. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
So I hope what I've convinced you is we've gone in really just about 2 decades from the vaguest idea of what genomic medicine was going to be all about. And we've started to bring clarity. It's in focus. I think there's far more that's going to come that's going to be exciting than we've seen so far. But what we've seen already is truly, I think, inspiring and really is in many ways fulfilling the promise of the Human Genome Project. And what's been wonderful about this, and it's only been possible because, again, we went from this very small community of scientists that were able to accomplish the Human Genome Project, and now 35 years later, the realization of genomic medicine is only because of this incredible community that I get to represent and we support through grants and so forth to see their accomplishments. So with that, I will stop. If you enjoyed what you heard and you want to stay in touch, we do all sorts of things. First of all, we have an incredible website. And one way to sort of navigate it is if you go to that Stay Connected URL at the top. I do various things with my communication group. I send out a monthly newsletter that you can subscribe to if you want. If you want to get an extra email a month, I keep people updated. And for those of you who like social media, I am on 3 platforms at the moment, probably heading to the 4th, but including most recently Instagram, which I've been told makes me cool. So that's what I've been told. And with that, I'm going to stop. And I think many of you are going to come up and we're going to have questions. Thank you very much.
Thank you so much, Dr. Green. So before we pass out to Q&A, the audience, I'm just going to ask you a few questions, if that's okay.
Okay, sure.
So how have Jewish values influenced your career in this research?
So, one of the things I would absolutely say is one of what I have appreciated about genomics, and it certainly matched my personality and my upbringing and so forth, is this incredible sense of enabling others to be successful and of sharing. I mean, again, there's so many sub-stories I could tell, but the other ground, there's many legacies of the Human Genome Project. But one of them, which I didn't happen to mention, was that that the whole concept of sharing scientific data, that scientists traditionally didn't share well. They were like kids. They just didn't share. They didn't want, and the Genome Project completely busted that mold completely in that they started to share the genomic data through the internet prior to any papers, prior to any credit. It just got shared. That is now promulgated across an entire cultural change in science, such that all scientists now must share data much more aggressively. And the Genome Project really is to credit for that in environmental science. So I think that's, and then even so many of these aspects that you see, these these attempts to just knock these barriers down, and even the fact that our institute, our whole goal is to get genomics everywhere else. We don't want to hold it to ourselves. We want everybody else to. So I think there's a sharing and enabling that is a sort of the core values. Not to mention fairness, equity, or a lot of things, social justice. There's a lot of stuff. Oh, let me tell you one other thing, because it sort of spoke to the Genomic Society. I didn't actually even use the word, but I probably should. Another very different thing about the Human Genome Project is from the very beginning, they dedicated a portion of their money to studying the ethical, legal, and social implications of what they were doing. The reason I want to spell that out is you may hear the phrase ELSI, because it became a coined word. ELSI, ethical, legal, social implications. ELSI research is now a coined phrase, which basically is bioethics. And so even today, we spend 5% of all of the money we do is spent to support bioethics research or ELSI research, which isn't terribly expensive because it doesn't require laboratories or computers. And it's a different kind of research. So we are the leaders in bioethics. Our field of genomics is the leaders of bioethics research in bioethics.
So I was going to ask about ethical concerns, but I guess that covers that. So maybe just selfishly, because I'm involved in AI research.
Yes.
What AI genomics research is coming down the pipe?
Oh wow, yeah. No, besides fun PowerPoint slides, I would certainly want to. No, AI. It's incredible. In some ways, often it's. Although I'm not an AI, you're much smarter than I am around AI, and I'm too old to really even fully understand it. But in so many ways, it's made for each other, right? Because so much of what's going on now in genomic medicine, first of all, trying to, taking in volumes of information, huge amounts of information about human genomic variation and phenotype or, characteristics of people and their medical records and trying to develop correlations. Human brains just can't do that. You need AI-like approaches at every level along the way. Massive amounts of it. I mean, the way we're going to really see genomic medicine advance is going to be hugely big data. Massive data studies, millions of people, their genome sequence, electronic health records available, lots of other studies on them. And the data analysis is rapidly becoming the barrier. AI is going to fit into this. And we are supporting many, many avenues of research of using AI.
In the first couple of years of medical school, every time they mention, do you know who's most susceptible to this disease? And then the class has a chorus together, Ashkenazi Jews. Yeah. So, can you speak about some of the advancements or maybe some of the research that is particularly impactful for this community that's coming?
Well, I think first of all, this is a community that has appreciated and has been. I mean, actually, it's sort of funny when people have started saying, "What was your very first genetic test I ever had?" It was when we were considering having your classmate, my son. We got Tay-Sex screen. I mean, even though it wasn't even a DNA-based test then, now it's DNA. Back then it was an enzyme, but it was this proactiveness of appreciation that this is something that we absolutely can do something about with intellectual advances and social advances and organize this in a way that is a model for how this can be done for other communities and other diseases. So absolutely, this is a community that has always been at the forefront of implementing these sorts of things.